Thank you for tuning in. My name is Anna, and I'm a graduate student at the University of Kansas, and my thesis focuses on jellyfish venoms. I'm pretty excited to talk about the results of this project on pore-forming toxins that are likely most well known as some of the most potent toxins in the venoms of box jellyfish. Now, these aren't box jellyfish here. These are moon jellyfish or Aurelia that we have in the lab, fairly weak stingers. And they're not the focal species of this project, but I will talk about them a bit near the end. So jellyfish and their relatives are part of the phylum Cnidaria, which is the earliest diverging venomous clade. On the right here, I have a simplified tree of some of the major Cnidarian groups, and my project really hones in on the Medusozoans, or the jellyfish-bearing lineages. Now this includes cubozoans, or box jellyfish, scyphozoans, or true jellyfish, starozoans, or stalked jellyfish, and the hydrozoans. Now the venoms of Cnidarians are primarily used to subdue prey or deter predators. But as many of you may be familiar, some species of Cnidarians are capable of stinging humans. And there's a back-of-the-envelope calculation that about 150 million stings occur every year worldwide due to Cnidarians on humans. Now, despite their medical relevance and their importance in ecological interactions, the venoms of most Cnidarians are poorly understood. But one family of toxins that is fairly well known is the aptly named jellyfish toxins, or JFTs. Now, this is a family of potent pore-forming toxins that are Cnidarian specific and were initially isolated from box jellyfish. Now, if you're unfamiliar with this species here, this is Chironex fleckeri, or the Australian box jellyfish, and this is widely renowned as one of the most venomous animals on the planet, at least towards humans. Those tentacles can get several meters long. 2 meters of tentacle contact with this animal is enough to kill an adult human in 2 to 3 minutes from cardiac arrest. And that's largely been associated with the presence of these JFTs within the venom, which are highly hemolytic and cardiotoxic. Now, the bioactivity of 7 JFTs has been characterized, some from Chironex, as well as 3 other species of cubozoans, so 4 total. But as more sequencing data has become available for a wider diversity of Cnidarian species, more JFTs have been identified, though not characterized at the bioactive level or the protein level. So our question was relatively simple. What is the evolutionary context of JFTs? And can we use this to better understand Medusozoan venom evolution more broadly? So we split this question into 3 pieces. How widespread are JFTs across Cnidaria? How are these JFTs related to each other in a phylogenetic context? And if we have a good understanding of those relationships, can we use molecular selection analyses to help us better understand why it is that box jellyfish JFTs are so potent compared to others? Now, I'm going to be talking about the molecular selection analyses a bit later on, but just for the sake of time, I'm going to go right into the results from these first 2 questions. So if you are curious about the methods for both of these, in particular the bioinformatic pipeline that I used, I'm happy to talk about it either in the chat or after this presentation. So jumping right in, this is the final maximum likelihood tree from the NMI analysis. There's a few things I want to point out before I talk about the results. You're not going to be able to see the species names of each of these sequences, but I have color-coded the tree for those major lineages that I talked about earlier. So red is for box jellyfish or cubozoans, green is for true jellyfish or scyphozoans, and blue is for hydrozoans. Now, wherever you see a circle within the tree, that represents 100 bootstrap support, and we had fairly good support throughout the tree. The last thing I want to point out is these sequences here uses the root. This is a group of bacterial endotoxins called cryotoxins that show the strongest secondary homology towards JFTs and have been used in previous studies to root other phylogenetic analyses. So I used 147 publicly available transcriptomes across Cnidaria to search for JFTs, and I found 111 novel toxins across Medusozoans. We did find some toxins in anthozoans, but they placed poorly within the tree. There's a main split within this tree. So JFT1, which are the sequences represented up here, and JFT2 down here. Now I should note that most of the previously annotated JFTs actually fall within JFT1. The other thing to note is that all those functionally characterized cubozoan toxins also fall within JFT1. Most of the sequences in JFT2 are novel to this study. So I want to hone in on JFT2 for just a moment. So we split this into 3 subclades. So JFT1A contains 2 sequences from a coronate scyphozoan Atolla. JFT1B is cubozoan specific, and JFT1C is a mixed taxonomic clade. So there's some box jellyfish toxins, some hydrozoan toxins, and then several species of true jellyfish represented. So this JFT1b, this cubozoan specific group, contains 35 sequences of JFTs. So this is one of the largest taxon specific clades within the whole tree. But there's actually only 6 species represented here. So that suggests to us that there's a pretty high level of duplication occurring within this group. The other thing I want to note is that there's a split within the subclade. So what has previously been called type 1 JFTs and type 2 JFTs. This is based on previous studies on this group that really focused just on these cubozoan JFTs. So this was pretty reassuring to us that even though our sampling is much larger, we're recovering some of these previously shown relationships of JFTs within just that specific group. So now I want to talk a bit about the molecular selection analyses that we used. So all these tests were conducted in HiFi, and the tests I'm showing here are either for episodic positive selection or relaxation of selection. They're also either gene specific or branch specific tests. We did do some site specific tests, but they're not shown here. Now these tests can be run in two flavors. One is where you set a subset of your branches as the reference towards a test, and so these are comparative tests. Another is an exploratory test, where you actually search through all the sequences in your data set to do the analysis. So for these tests, I did JFT1 versus JFT2 and vice versa, as well as an exploratory test. But because we had such an interesting kind of relationship within JFT1, I also did JFT1b, this cubozoan specific group, versus JFT1c, vice versa, and then an exploratory search using these tests. We didn't include the Atolla sequences for these. So overall, the results were mostly not that unexpected. Using HiFi to conduct this DNDS, to calculate DNDS, most of these were below 1, which was not unexpected for toxins, especially from early divergent groups such as Cnidarians. We found in our comparative tests for relaxation of selection, always found evidence of relaxation of selection. That's also not that unexpected. In our branch-specific tests, we did find branches that were under episodic positive selection, but many of these were just single branches. They weren't that deep within the tree, so not that exciting. Where it did get exciting was with this test BUSTED, which is a gene-wide episodic test of positive selection, which is looking for at least one site in at least one branch of your test branches. Because it's looking for just at least one site in at least one branch, it's not that unexpected when we're looking at these much broader groups of JFT1, JFT2 in the total, that it finds evidence for positive selection. But it does get exciting when we look just in these tests in the JFT1 group. So when you're searching JFT1b, this cubozoan group, as the test with JFT1c as the reference, you find evidence of episodic positive selection. But when you do JFT1c as the test with JFT1b as the reference, you do not find evidence of positive selection. So what we've done here is we've really narrowed it down to say that this group, the cubozoan specific group, where we know we have these potent pore-forming toxins that have been previously characterized, is showing evidence of episodic positive selection. And this was pretty exciting for us to see. So the conclusions. So JFTs are much more widespread across medusozoans than we previously expected. And importantly, JFTs are not just present in these medically relevant species. They're even in some species that we consider harmless or weak stingers, like Aurelia. So we did know this duplication in the cubozoans. I talked about that earlier. There's also several JFTs within Aurelia as well. So it'll be interesting to see if those can be bioactively characterized, if these also show this potent pore-forming bioactivity. We found these distinct taxonomic patterns emerging through our phylogenetic analyses of these JFTs. And because we had a good understanding of these relationships, we could do the molecular selection analyses. And what it showed to us is what we think is happening is in these box jellyfish JFTs, they've undergone extensive duplication of these JFTs, followed by episodic positive selection that may be selecting for greater potency on potentially this targeting of blood cells or cardiac cells that may be making their JFTs much more potent and dangerous for humans as opposed to others. And this is the first time this has been described within this group. So it's very exciting for us to see. So where am I going from here? So like I said, I'm at the University of Kansas, so I'm not going to be doing any field work with box jellyfish. But what we do have in our lab is this species, Hydractinius symbiolongicarpus. This is a colonial hydrozoan, and I found 6 JFTs associated with this species that I can find throughout my maximum likelihood tree. And unlike the majority of the species represented here, the genome of Hydroctinia has been annotated and well sequenced, and we have a variety of transcriptomes available. The other really nice thing about Hydroctinia is it's pretty amenable to genome manipulations techniques such as CRISPR and short hairpin gene silencing. So I'm using both of those techniques right now, both the CRISPR and the short hairpin, to knock out each of these JFTs and look for a phenotype. We're expecting that these JFTs are important for prey capture in the species. So after these knockouts, I'm testing for their ability to capture prey using feeding assays. And I'm hoping to have some of the results for those at a future SICB. So I want to say a huge thank you to both my academic and my financial support. This project has been going on for a few years now, and I'm pretty excited to be able to present the results today. And a thank you to the SICB DEE Student Presentation Committee for inviting me to talk about this project. And I'd be happy to answer any questions or get any feedback for this. Thank you very much.